Hello everyone, myself Dr. Suresh, Professor of Biochemistry, and in this video I'll be teaching you the digestion of proteins and their absorption. So to talk about proteins, so in our earlier videos we have already discussed the chemistry part of proteins and amino acids. So we're all aware how proteins are made, right? So the building blocks of proteins are amino acids. So whether they are from outside or from inside, their basic building structure is amino acid, right? So there are 20 different types of amino acids do exist in our body. So out of 20, 10 are essential amino acids and 10 are non-essential amino acids. By the definition, we are all aware. Essential means the amino acids which cannot be synthesized in the body, but they are supposed to be supplied through diet. But in case of non-essential amino acid, yes, they do exist in the diet, but the other facility is they are also can be synthesized in our body from various chemical reactions. So that's why if you are not providing them in the diet also, no issues, as our body cells are capable of synthesizing these amino acids. So to go in detail about the digestion part, so few basic aspects we have to discuss, like the main role of amino acids. Before going in detail about the proteins, we should know the basic aspects of amino acids, right? So the main role of amino acids in the synthesis of structure and functional proteins. As I earlier mentioned, basic unit of proteins amino acids. So amino acids are must for structural as well as functional part of proteins. And also a 70 kg man, okay. So the normal protein turnover is 400 grams per day. Okay, what is that? A 70 kg man, in a 70 kg man, daily protein turnover is 400 grams per day. That means the same amount of protein will be synthesizing, at the same time the same amount of protein will be degrading. So the rate of synthesis of protein is always has to be equal to the rate of degradation of protein. So they should be in balance. No one process has to be imbalanced, right? And another aspect is essential amino acid has to be provided in the diet, because lack of a single essential amino acid, or otherwise you can say out of 20 amino acids, if there is missing of single amino acid, there is no continuation of protein synthesis, right? And even the body amino acid pool, there is a pool called amino acid pool. So what happens whenever there is a degradation of protein, okay, the degraded part of proteins are amino acids. So these amino acids go and accumulate in the pool. So based on the demand, these amino acids will be supplied. Same way, when you had a food or the diet of containing of proteins, when they get digested, so first they will go to amino acid pool. From there they will be distributed based on the demand, right? So as I mentioned, rate of synthesis of proteins always equal to rate of degradation of proteins in our body, right? So that the nitrogen balance has to be maintained. Why nitrogen? Because the main source of nitrogen in our body are proteins. That's why proteins are made up of amino acids, amino group, which is having nitrogen, right? And one more important thing, proteins are not mainly for energy, they are for structural and functional aspects. Remember, like other biomolecules, carbohydrates and lipids, they are both involved in production of energy mainly, but proteins are not like them, and they are purely for structural and functional aspects. Okay, so when you see the diagrammatic representation here, so there is a body amino acid pool which will be maintaining like 100 grams, right? So what are the sources to maintain this body's amino acid pool? Dietary proteins, like which will be contributing 50 to 75 grams to the 100 grams of amino acid pool at the same time, and majority portion 300 to 400 grams will be coming from breaking down of body proteins. So now this is the contribution towards the amino acid pool, and from the amino acid pool, how they will be distributed. Okay, these amino acids, the pooled or the accumulated amino acids, how they will be distributed. So one is from the amino acid pool, the amino acids will be converted to keto acids. Okay, so the keto acids will undergo oxidation, otherwise gluconeogenesis, and also in synthesis of non-essential amino acids, right? And when they are converting into keto acid, these amino acids will remove their amino group, and that amino group will be ammonia, and ammonia converted to urea, which will be excreted. So almost 15 to 30 grams of nitrogen in the form of urea will be expelled out of the body. So synthesis of specialized products, heme, purines, pyrimidines, creatine, enzymes and hormones. Why? Because in purines and pyrimidines, they are the nitrogen bases of nucleotides, and for their synthesis some of the amino acids required in the way, and like their carbons and nitrogens may required for making of purines and pyrimidines. So this is an overview of metabolism of amino acids before talking in detail about the digestion of proteins. So what are the enzymes which play a major role in digestion of proteins? Because the proteins that we are consuming, so in olden days, I mean like raw protein we used to consume, but nowadays because of the cooking methods and as there is a change in lifestyles, we are all addicted for to have steamed or cooked items. So same way, the main source of proteins is non-vegetarian sources, meat, red meat or like chicken, right? And they do exist protein sources in vegetarians like pulses, cereals, right? Then how to digest? In chemistry part of proteins we have discussed denaturation. Denaturation is nothing but disturbing the secondary, tertiary and quaternary structure of proteins, by the way, and primary structure we cannot disturb as there is a strong bond exists, peptide bond, right? So denaturation may be of two types, physical and chemical. So cooking is nothing but denaturation of protein. So what is happening here? The cooking causes denaturation of protein so that they can be easily digested by our body enzymes and they can easily be absorbed, right? Cooking is nothing but heating. So heating is also one of the physical agent which causes denaturation of protein, right? And apart from that, they do existence of proteolytic enzymes, the enzymes which acts on the proteins for the degradation, known as proteolytic enzymes, and initially they will be secreting as inactive form. Okay, the inactive form of proteolytic enzymes is secreted as zymogens, it is inactive form or inactive enzyme, and they will be converted into active form in the intestinal lumen. Okay, so the main thing with the proteolytic enzyme is this would prevent autodigestion of secretory acini. That means if directly they will be produced as active forms, there is a chance of autodigestion, eating of own cells. So that's why, because cells also have protein content, right? So premature activation of trypsinogen results in acute pancreatitis. So the result of autodigestion, because trypsinogen is a proteolytic enzyme, so this will be producing from the pancreas. So if trypsin, what happens? Trypsin is an active thing, trypsinogen is a inactive form. So instead of trypsinogen, if pancreas is producing trypsin, it starts eating the pancreatic cells and causes acute pancreatitis. So it is which is a life-threatening condition, and you might have all aware of the famous founder of Apple who has died with the acute pancreatitis, right? So the proteolytic enzymes are the enzymes which mainly involved in digestion of proteins. So here there are two types of proteins existing in our body. One is endopeptidase, the other one is exopeptidase. So here endopeptidase means these are the enzymes which acts on a protein in the middle of the protein. Like if you say, if a protein is made up of 100 amino acids like 1, 2, 3, 4 and 5, right? It is having two terminals, amino terminal end and carboxy terminal end. So here endopeptidases will work in right middle of the protein. So the protein will be converted to shorter fragments of polypeptides. So that's what endopeptidases will do. Okay, so the category of enzymes which come into endopeptidases like pepsin, trypsin, chymotrypsin, elastase. So remember, so they break lengthier proteins into smaller polypeptides, okay, and shorter polypeptides. So they cannot liberate free amino acids. So endopeptidase cannot liberate free amino acid. Now coming to the second category, that is exopeptidase. So exopeptidases, so they can act only on the peptide bond located at the ends. So what I told you, like they do existence of two terminals in a protein. One is amino terminal end, other one is carboxy terminal end. So these exopeptidases will work on any one of the terminal side of the protein or again shorter polypeptides, right? So based on the terminal side they are working, they are named as carboxypeptidases and aminopeptidases. Carboxypeptidases, what they do, carboxypeptidases will be working on carboxylic end of the peptide or polypeptide, and aminopeptidases which are acting on the peptide bond which is next to the amino group, right? So this way they will work. So to summarize, two types of proteolytic enzymes in our body for digestion of proteins, endopeptidases and exopeptidases. Endopeptidases, right in middle of the protein they will act and they break the protein into shorter polypeptides and peptides, And whereas exopeptidases, they will work on only one peptide bond which is existing next to the any one of the terminal, amino side, amino terminal side or carboxy terminal side. So based on that, names are there, carboxypeptidases and aminopeptidases. So here diagrammatic representation you can make out here: proteases, exopeptidases and endopeptidases. You see here endopeptidases, they are working on the right middle of the protein, okay, and the resultant products will be shorter peptides, shorter form of polypeptides and peptides. And if you see the way of mechanism of action of exopeptidases, you see aminopeptidases, so which is working on the amino terminal side. So there is a chance of liberation of one free amino acid from the broken protein parts. Okay, now dipeptidyl peptidase. So what is dipeptidyl peptidase? So it will work on the from the one side of the amino terminal end, right, two amino acids. So it will liberate two amino acids with a single peptide. And tripeptides, like it can liberate a tripeptide, okay, with two peptide bonds. Carboxypeptidase, I told you, it will works on the carboxy terminal of the peptide to release free amino acid. And peptidyl dipeptidase, which can work on carboxy terminal side of two sets of amino acids which are in the peptide linkage. And dipeptidases, which work on the amino acids which are in the peptide linkage, so it will release two free amino acids. Tripeptidases work on the peptides which are having two peptide bonds and releases one free amino acids. Omega peptidases, which work on a continuous form of shorter polypeptide chains and releases set of two amino acids at a time having one peptide bond. So this way exo and endopeptidases will work. So to start off with the digestion of proteins, okay, as you all aware, the digestion part will start in the mouth when the food is mixed with the saliva. But unfortunately, there is no proteolytic enzyme that present in the mouth, so the food straight away goes to the stomach. So in the stomach, there are chemicals like chemical solutions which are present like HCl, hydrochloric acid. So we all learned denaturation. It may be of physical, it may be of chemical. So chemical means concentrated acids, HCl, H2SO4. Okay, so HNO3. So all these things will cause denaturation of protein. So HCl is one of the chemical substance which mix up with the protein substance and it causes denaturation. That means it breaks the secondary, tertiary and quaternary structures and only primary structure is intact in the protein. So that's what it is making the work easy for proteolytic enzymes. And there is one more enzyme, pepsin. Pepsin is a endopeptidase which work in middle of the protein and breaks the protein into smaller peptides, right? And pancreas, which releases trypsin, chymotrypsin and elastase. So here you one thing to remember. So all these proteolytic enzymes, first they release as inactive form as pepsinogen, trypsinogen, chymotrypsinogen and proelastase. Okay. So here pro and zymogen both are like inactive forms. Okay. So along with HCl, pepsin will cause the digestion. I mean half the digestion part will be completed in stomach. So from stomach, the food will reaches to the upper part of duodenum. There the pancreas releases some of the watery solution like with bicarbonates and in combination of trypsin, chymotrypsin and elastase. So they will complete the other half of the digestion of the proteins. So in intestine, when lower parts of the intestine the food is reaching, so there carboxypeptidases, aminopeptidases, dipeptidases which completes the digestion part of proteins and they form individual amino acids. So to study in detail about the digestion part in the stomach, as I told you, hydrochloric acid is secreted. It makes the pH optimum for action of pepsin and also activate pepsin. Pepsin is a acidic enzyme. Remember, because HCl is acidic in nature, so we have all learned factors affecting enzyme activity. So the pH also plays major role in enzyme activity. So being acidic enzyme, pepsin can play a major role in digestion of proteins when there is a HCl. Okay. So the acid also denatures proteins, as I said. Renin, this is one more enzyme which excreted from stomach and this plays major role in case of newborn babies. So the main protein present in milk, that is casein, and this casein has to be acted by renin. Okay, the other way, the other name of renin is chymosin. It active in infants and is involved in curdling of milk. And with the age, it will get vanished. So it is absent in adults. Milk protein casein is converted to paracasein and by the action of renin. This denatured protein is easily digested further by pepsin. So before the action of pepsin, there is action of renin. So pepsin, so how pepsin is secreted? Pepsin is secreted from chief cells of stomach as inactive pepsinogen, as I mentioned earlier. The conversion of pepsinogen to pepsin is brought about by removal of 44 amino acids from N-terminal end by the hydrochloric acid. That means inactive form to active form. Okay, so here the activator is, so pepsinogen is in inactive form converted to active form pepsin. So here the activator is HCl. Okay, and once pepsin is activated, so it will do its function by degrading the protein. So the optimum pH of activity of pepsin is around 2, so acidic. So pepsin is an endopeptidase, as I told you, it works in mid of the protein part, and pepsin catalyzes hydrolysis of bonds formed by carboxyl group of aromatic amino acids phenylalanine, tyrosine, tryptophan, and sulfur containing amino acid methionine. So by the action of pepsin, proteins are broken into proteoses and peptones. So they are the shorter fragments of proteins. So renin, this also we have already discussed. Renin is active in infants, as I told you, and it involved in curdling of the milk. It is absent in adults. The milk protein casein is converted to paracasein by the action of renin. So one key note to remember here, renin, the spelling you see double N and single N. So there is a difference. Double N renin is a proteolytic enzyme present in gastric juice, and single N, that renin, is a proteolytic enzyme secreted by kidneys. It is involved in activation of angiotensin mechanism. So okay, so renin angiotensin mechanism, which is a hypertensive agent in production of angiotensin, which is a hypertensive agent. So the optimum pH for activity of pancreatic enzymes is 8, provided by the alkaline bile and pancreatic juice. So what are the conditions in stomach? Acidic conditions, right? But pepsin, okay, being acidic enzyme, it is acting perfectly. But the food, when it is mixed up with the acid, it is a chyme. When it is coming to the upper part of the duodenum, so the upper part of the duodenum, it cannot handle the acidic part of chyme. So it has to be neutral, and moreover the enzymes which are coming from the pancreas for the next digestion part of proteins, they are alkaline in nature. Then how they do work on acidic chyme? So to make it work, pancreas first releases watery secretion to neutralize the acidic chyme. That means that watery secretion contains bicarbonate ions which neutralizes the acidity. So once acidity neutralizes, this proteolytic enzymes like trypsin, chymotrypsin, so elastase, all these will come into the action and they will finish the other part of the digestion of proteins. Okay, and the secretion of pancreatic juice is stimulated by peptide hormones. Two peptide hormones are there, cholecystokinin and pancreasamine. When there is a acidic chyme in the upper part of duodenum, it is like stimulus to secrete these hormones, cholecystokinin, pancreas. What they do, once these cholecystokinin, pancreasamine activated, they stimulate the biliary duct to secrete bile, okay, and also pancreas to release the proteolytic enzymes. So pancreatic juice contains important endopeptidases, namely trypsin, chymotrypsin, elastase, and carboxypeptidase. Remember, they all will be released as inactive form, that are such as zymogens like trypsinogen, chymotrypsinogen, and proelastase. And once for trypsinogen, what we require? We require trypsinogen by autocatalysis. Once the trypsin has to converted into trypsin, by autocatalysis further trypsinogens will be converted to trypsin, and also not only autocatalysis, this activated trypsin will activate chymotrypsinogen to chymotrypsin, proelastase to elastase. So all three serine proteases, the active center of these enzymes contains serines. So all these proteolytic enzymes, they have active centers, like what they contain, active centers, they are made up of serine amino acid, serine residues. Trypsinogen is activated by enterokinase, as I told you. So first trypsinogen has to be activated with the help of enterokinase. Once one trypsinogen get activated as trypsin, trypsin activates further trypsinogen to trypsin, and also it activates further proteolytic enzymes such as chymotrypsin, chymotrypsinogen, and proelastase. So acute pancreatitis, what is? So premature activation of trypsinogen inside the pancreas itself will result in autodigestion of pancreatic cells, results in acute pancreatitis. Okay, and it is a life-threatening condition, so person may not survive for long. So trypsin will act on chymotrypsin such manner the A, B, and C peptides are formed. Those three segments are approximated so that active site is formed. The selective proteolysis produces the catalytic site. Trypsin and chymotrypsin degrade the proteins into smaller peptides. These are further hydrolyzed into dipeptides and tripeptides. by carboxypeptidases which are present in pancreatic juice. The pro-carboxypeptidase is activated by trypsin. They are metalloenzymes requiring zinc. So metalloenzymes are, all enzymes are proteins, right? So proteins also, they are require some metals to get activated. So metalloenzymes, for their activity, they require inorganic substance such as metals. So one among such a metal is zinc. So pro-carboxypeptidases, they are all not pro-carboxypeptidases. So all pancreatic enzymes, they require zinc, and zinc abundantly present in pancreas. So here you see I am showing you the tabular column here. So which of the endopeptidases acting on which, like if there is a protein combination, like all 20 amino acids are there. So we already studied the classification of amino acids: simple amino acids, aromatic amino acids, branched chain amino acids, sulfur containing amino acid, basic, acidic amino acids. So all like classification we have studied. So here pepsin being endopeptidase, it will work on right middle of the protein, where so there should be a signal or there should be a mark where the pepsin has to go and attack the peptide bond. So here in middle of the protein, where there is a rich quantities of tyrosine, phenylalanine, tryptophan, okay, and glutamic acid. So when there is a rich sequence of these amino acids are there, pepsin will go and break the peptide linkage. Same way trypsin, where there is a rich quantity of arginine glycine. Okay, if this is a protein, if there is a rich quantities like side by side arginine glycine, here trypsin will come and breaks the protein into two parts. Okay, same way chymotrypsin, where there is aromatic amino acids are there it will go and it will break the protein. Okay, and elastase, neutral amino acids like glycine, serine, alanine. Okay, and carboxypeptidase, exopeptidases, where tyrosine, phenylalanine, tryptophan, valine, leucine, isoleucine, and carboxypeptidase B, like basic amino acids, arginine and lysine, then it will break up. Complete digestion of small peptides to the level of amino acids is brought out by the enzymes present in intestinal juice, succus centricus. The luminal surface of intestinal epithelial cells contain following enzymes. Aminopeptidases releases N-terminal amino acids. Dipeptidases and tripeptidases will complete the digestion of protein further. So here in a tabular column, I am showing you the enzyme and the source of these enzymes and where they present and what are all the activators required for the activation of these enzymes and which bond they specifically acting on and what are the resulted products. So first to talk about pepsin. Pepsin is endopeptidase, okay, and it is present in gastric juice, activators HCl to convert pepsinogen into pepsin, and which is working on it being an endopeptidase, it is working right middle of the protein, and peptide bonds formed by carboxyl group of aromatic amino acids and sulfur containing amino acid methionine, and resultant things are proteins will be converted to shorter forms proteoses and peptones. And renin, gastric juice, it is helpful in curdling of milk, okay, then only this casein will be digested properly. And trypsin, it is also endopeptidase which is present in pancreatic juice. Here activator is enterokinase, okay, and cholecystokinin is also which is involved in stimulation of pancreas to release this trypsin, and peptide bonds formed by carboxyl groups of basic amino acids, arginine and lysine. So resultant products will be proteoses, peptones and peptides. So remember, trypsin and other proteolytic enzymes releasing from the pancreas can able or capable to release the peptides. Chymotrypsin is also pancreatic enzyme. It is activated by trypsin, and it is acted by the peptide bonds like methionine, leucine and aromatic amino acids. And elastase, which is also endopeptidase, is also pancreatic. Activator is trypsin. Carboxypeptidase is also pancreatic juice enzyme. It is also activated by trypsin, and which is where it is acting. It is acting on C-terminal amino acid, free amino acid. So carboxypeptidase is a exopeptidase and which can capable of releasing free amino acid. And aminopeptidase also are present in intestinal juice, succus centricus, and they are also acting as aminopeptidases which release free amino acid, and tri and dipeptides also present in intestinal juice by succus centricus, and the peptide bond, they work on peptide bonds of small peptides and they are also capable of releasing free amino acids. So that's all about digestion part of proteins. Thanks for watching. Thank you.